Okay. So good evening, everyone. Today I am going to talk about protein synthesis. After transcription process, we have seen the genetic code and vowel hypothesis. So the next part is in the central dogma we have started with the DNA replications. Then we have seen transcription process. Then we have seen genetic codes and vowel hypothesis, and now it's translation process, which is also called as protein synthesis. The literal meaning of translation is when you are going to change the language. So if we'll see, this is the basic mechanism, this is the basic process, especially in case of eukaryotic cells. In our case we are having a cell plasma membrane. Inside we are having a nucleus where we are having our DNA. So inside the nuclear membrane our DNA is there. That DNA, whenever we need to cell divide, that time it is going for duplication. So cell replication will be, DNA replication will be there at the time of cell division. But cell division is a different part, and what we are going to talk about transcription and translation, that is a different part. Cell division occurs for complete DNA, and that is only at the time of when cell need to divide, cell need to replicate. Transcription and translation process are mainly for protein synthesis, and that is not for complete DNA, it is for only a specific portion of the DNA. Okay. So in case of eukaryotic cell, this transcription process occur inside the nucleus. Then mRNA will be, we are going to get mRNA, which is going to be moved to the outside the nucleus into the cytoplasm, and there ultimately this translation process is going to happen. So first process is translation. It is going to give you protein. That protein is going to be folded in a specific manner, in a specific pattern, that is called as protein folding, or you can call it post translational modification. So normally protein, once you are going to synthesize protein, that proteins are non-functional. It is in the same way as when you are going to produce a messenger RNA. This messenger RNA is of called as primitive messenger RNA, you can call it precursor messenger RNA. So this is non-functional messenger RNA. Once it is going to be processed by splicing, capping and tailing, then only you are going to get a functional messenger RNA. In the same way, once you are going to produce a protein, that protein will be non-functional till this protein will go for a specific folding and modification process. So this is overall mechanism of how it is going to happen. We have seen we are having DNA. So DNA is going to be transcribed into the messenger RNA. In the messenger RNA we are having a triplet codon. That triplet codon is going to specify amino acids, and ultimately we are going to convert messenger RNA into amino acid. So I was talking about the translation word. If you will see the message, the codes of DNA is adenine, thymine, cytosine, guanine, that is nucleotides. The language of this messenger RNA is also adenine, uracil, cytosine and guanine, means nucleotides only. So the alphabet of DNA and alphabet of RNA is same. So translation, we are not, means in translate, when you are going to do the transcribe, that time normally we are not going to change any languages. It is nucleotide to nucleotide only. But when you are going to convert this messenger RNA into protein, so here we are having the language of nucleotides, and we are going to convert them in the language of amino acids. So conversion of one language to other language is called as translation. Normally when you are going to convert Hindi to English, Hindi to Sanskrit, Sanskrit to Hindi, Tamil to Telugu, so means conversion of one language to other language is called as translation process. So here also we are going to convert a nucleotide language into the amino acid language, and that is called as translation process. If you will see the translation process, basically we are having 5 different steps. First step is the called as activation of amino acid, means directly amino acid does not go for protein synthesis. we need to activate the amino acid so that it will be properly involved, properly go and participate in the protein synthesis process, translation process. So that process is called as activation. Then we are going to initiate, elongation, termination, and then the modification of the protein, that is folding and chemical modification, structural modification, that is called as post synthetic processing or post translational modification. Post means after, translation means translation, and modification. So after translation we are going to do the modification. It is called as post translational modification. In this case, when we are going to talk about this translation process, we require certain things. So for all the processes, either in case of transcription or translation or duplications, we require certain components. So here also we require certain things needed during the protein synthesis. The most important thing is we require one messenger RNA, because here we are having a message on which protein synthesis is going to be formed, means where we are having a code for protein synthesis. So the messenger RNA, that is the specific information for your protein synthesis, is present in the messenger RNA, and according to the sequence present in the messenger RNA, we are going to arrange the amino acids, and that ultimately gives us the protein. So that is the significance of messenger RNA. Then second thing we require, amino acids. We know that we are having 20 important amino acids. Can anyone will tell me how many amino acids we are having in our body? In human body, how many amino acids we are having? Anyone can tell me?
So 20 only.
20 only. Okay. Anyone else?
So yesterday you told us about two more proteins, two more amino acids.
Okay.
So 22.
Okay, 22. Anyone else? How many amino acids do you have in your body? Pardon?
So 22 only yesterday you told.
Okay. It is not necessary that everything whatever I will tell it is correct only. I am going to change that word.
So but still I read about it after that.
So it is 22 only, right?
Yes sir.
Okay. Now correct that statement. If my question is how many amino acids we are having in our body?
Sir, 22 types, but not exactly 22 numbers.
I am asked. Okay. Again, so how many different types of amino acids we are having? This question is correct, Sanjit?
Yes, sir. So 22.
No. We are having more than 300 amino acids, 300 plus amino acids in our body.
The protein, the amino acid which form the protein are 22.
So I didn't ask that question. Means when I ask you that question, that time I ask you how many different types of amino acids we are having in our body. I didn't talk anything about protein synthesis.
Yes, sir. So proteinaceous and non-proteinaceous both are about 300.
So in our body we are having more than 300 plus different types of amino acids. Just a brief information about the amino acids. Any chemical molecules which is having amino group means NH2 or NH3 group, and acid group means COOH group. So any compound which is having amino group and acid group together in the same compound, we can call it amino acids. Clear? Out of those 300 plus amino acids, if you are going to see its structure, we are having different types of amino acids. We may have alpha amino acids, we may have beta amino acids, we may have gamma amino acids. So if you remember the amino acid structure, so roughly we are having one central carbon, one COOH, one hydrogen, one R group, and one amino group. Correct?
Yes, sir.
This is the basic.
Yes, sir.
This is the basic structure of any amino acid which normally we writing. But this is not the correct. This is the alpha amino acid. Why I am calling alpha amino acid? In the organic chemistry, whenever you are going to do the nomenclature, so the first carbon with whom COOH group, yeah, acid group is associated, because in organic chemistry this COOH group is the having the highest priority, yeah, most senior most group you can call it. Like in our family, if my grandfather is there, he will be the senior most, so first priority should be given to him. Yeah, you can call it, this is the Amitabh Bachchan of the organic chemistry. So wherever COH group will be there, you have to start from that side only. Okay, line will start from there only. So different type of nomenclature normally we are doing. One type of nomenclature is we are going to number carbon 1 2 3 like that one. So we will start and we are going to see the longest carbon chain. Forgot about those things here. So the first carbon with whom COOH group, yeah, acid group is associated, that is called as alpha carbon. So in this case, this is alpha carbon because this is COH group and this acid group is associated with this carbon. So this carbon is my alpha carbon. Correct?
Yes, sir.
And if alpha carbon have NH2 amino group is associated with alpha carbon, we can call it alpha amino acid. So in this case, this is my alpha amino acid, and whatever 22 amino acid, yeah, 20 amino acid, whatever we are going to see in the protein synthesis, they all are alpha type only. Again, it does not mean we are having only 22 alpha amino acid in my body. We may have number of other alpha amino acids also, but we use only those 22 different types of amino acids for protein synthesis. Why? What is the reason? Because we are having genetic codes which recognize only those 22 different types of amino acids. So we have only genetic codes for 22 amino acids. That's why those 22 special amino acids are going to be used for protein synthesis. So this is my alpha amino acids. I am coming there. Second, if I will write it, this is my carbon. So here we are having COOH group. This is carbon. So this is NH2 H H H R. Is this amino acid? Because according to my definition, any compound which is having acid group and amino group together in the same carbon, we are same compound, same case, we are going to call amino acid. So this is also amino acid, correct?
Yes, sir.
But the difference between the previous one which I have written here, if you will see, in case of second carbon, this compound, this is my alpha carbon and this is my beta carbon. So amino group is associated not with the alpha carbon, but amino group is associated with the beta carbon. Whereas in this case, this amino group is associated with the alpha carbon. So this is my alpha amino acid and this is my beta amino acid. Clear? Similarly, we may have gamma amino acid, we may have delta amino acid, depending on where amino group is associated. But all beta, gamma, though, other than those 22 amino acids, means we are not going to use for protein synthesis. So we require those 22 amino acids, 20 amino acids for protein synthesis. Again, here I am not going to consider pyrrolysine and selenocysteine because they are normally going to be chemically modified later on, or some specific case those are going to be used. So we are going to consider those 20 amino acids here. Any question? Is it clear?
Yes, sir. Clear.
In our body, we are again, just for your information, whatever 20 amino acids, we have different types of classification for those amino acids. We have 4 different way of classification. First classification of these amino acids we are doing on the basis of their R group. R group means on central carbon we are having a R group is associated. So we may have aliphatic R group, we may have OH containing R group, we may have sulfur containing R group, we may have acidic amino acid, we may have basic amino acids. So these are different on the basis of their R group. Then second classification is on the basis of their polarity. So we are having polar, non-polar, acidic, basic, 4 different types according to this polarity. On the basis of this one we are having 7 classes. Some other day we will see this one. On the basis of polarity we are having 4 types. Third way of classification is on the basis of its essentiality. Means amino acids are essential for me or it is non-essential for me. This essentiality depends on are you able to synthesize it or you are depending on external sources. If certain things I can synthesize in my body whenever my body needs, so they are called as non-essential, because if your body needs it, you can make it, you can prepare it, you can ready it, you can synthesize it. So they are called as non-essential. So in my body we are having 10 essential amino acids and 10 non-essential amino acids. Those amino acids which you cannot synthesize it, for those amino acids you depends on your diet, your food, your supplements, tablets. So they are called as essential, because it should be part of your foods. So they are essential amino acids. This is third classification. And fourth classification is on the basis of what it is going to be give you, glucose या carbohydrate or lipid. So it is glucogenic or ketogenic. Clear? This is clear?
Yes, sir.
Okay. So this is our amino acids. We require 20 amino acids. It may be polar, it may be essential, it may be non-essential, it may be any class, but those 20 amino acids we need. Then we require ribosomes. Ribosomes normally called as protein synthetic protein factory. I am coming there on the ribosomes. I will have a detailed informations. So we require ribosomes. Then we require transfer RNA. This is the carrier of this amino acids. So amino acid will not come means without vehicle it will not come. So amino acid require a carrier. Amino acid require a vehicle, a car, and this transfer RNA works as the carrier for this amino acid for protein synthesis. So wherever this amino acid will go, it will go loaded on the tRNA. So tRNA is a carrier molecule. Okay, for this one amino acid going to be loaded on tRNA. For that one it requires a specific enzyme. The enzyme which joins this amino acid on tRNA is the amino acid tRNA synthetase. So this is amino acid specific, means we are having 20 amino acid, so we require 20 amino acid tRNA synthetase. So amino acid tRNA is amino acid specific, means for every single amino acid we are having a specific enzyme, synthetase enzyme. So we are having 20 amino acids, means we require 20 amino acid tRNA synthetase. Then we require energy source. During the protein synthetic process, either it is initiation or activation or elongation, we require normally 2 types of energy sources. Some places we are going to use ATP as an energy source, some places we are going to use GTP as an energy source. So both ATP and GTP are required for the supply of energy during the protein synthesis. I have told you some places ATP, some places GTP, but most of the cases we require GTP. Then last part is protein factors. Like we have seen in case of eukaryotic cases, in the transcription we require certain factors, transcription factor during the transcription. Here we require, during initiation we require initiation factor, during elongation we require elongation factor, during termination we require termination factor. When we are talking about prokaryotic system, that time simple IF, EF and TF we are going to use the term. And when we are going to use eukaryotic system, that time a small e will be used as a prefix. So it is eukaryotic initiation factor, eukaryotic elongation factor, eukaryotic transcription factor, termination factor. So these are different protein factor we require during the course of protein synthesis. This is clear for everyone?
Yes, sir.
Any question anyone is having? Otherwise I will go for ribosomes. No question?
No, sir.
Okay. So I am going to ribosomes. We'll see some of the, because ribosomes is one of the important component for protein synthesis. So we will see some of the information on the ribosomes, and then we'll go for tRNA, and then we will initiate the protein synthesis. So first is ribosomes are known as protein factory of the cell. And which organ is known as protein factory of our body? Human body. Which organ of our body in our body called as protein factory of human body or animal body?
So endoplasmic reticulum?
No, I am talking about organ, not cell organelles.
The liver.
Okay, why?
So because most of proteins are formed in liver.
Yes, most of the proteins, maximum protein, because if you will see metabolically, liver is the one of the most important, metabolically most active organ of our body. Liver doing synthetic activity also, metabolic activity also, degradation activity also, xenobiotic biotransformation process also, hematopoietic process also, immunological process also, storage activity also. So number of types of activity liver is doing. So liver is one of the most important organ, you can call it, metabolically especially, and it is protein factory of our body. So in this case, ribosomes are called as protein factory of the cell. They functions as a reading messenger RNA, because I have told you on the messenger RNA we are having message generic codes. This code will be read by ribosomes, and it is going to translate those ribosomal code or messenger RNA codes and build proteins from those amino acids. Ribosomes are normally composed of two components, two subunits. We are going to call it large subunit and a small subunit. The main function of a small subunit is to read the mRNA codes. Remember this one. This may be one of the question, that which subunit of ribosome is going to read the mRNA. It is large subunit or the small subunit. The messenger RNA code will be read by a smaller subunit of ribosomes. Then what is the function of large subunit? Large subunit normally assemble and form the polypeptide chain from the amino acid. So a smaller subunit read this code, and large subunit joins the amino acid to amino acid and form the protein polypeptide chain. Each subunit, either it is smaller or it is larger, they are composed of some ribosomal RNA plus protein. So together they are called as ribonucleoproteins. So ribosomal subunits are composed of ribonucleoprotein, means ribosomal RNA plus proteins. These two subunits assemble to translate mRNA and disassemble when translation is complete. In a normal cells which does not participate, which is not going to do protein synthesis, that time, if this is the ribosomal two subunit, one small and second one is large, during non-translation stage, means when cell does not do any kind of protein synthesis, that time these two subunit will be separated. So large subunit is separate and a smaller subunit is separate. When cell need to do protein synthesis, that time messenger RNA comes, and first a smaller subunit will join, then large subunit will come, and then protein synthesis is going on. We are going to see those processes. So this is the mammalian ribosomes. Normally we are going to denote into this weight work unit S. So this is complete ribosomes. This is the large subunit, this is the smaller subunit of mammalian means or you can call it eukaryotic ribosomes. So the large subunit will be of 60s size and a small subunit will be of 40s size. When you are going to join them together, large subunit is going to join with a smaller subunit, it does not make 100. 60 plus 40 does not make 100. 60 plus 40 makes it 80 because some portions are overlapped with each other. Okay. Large subunit of eukaryotic ribosomal RNA subunit contains 3 important ribosomal RNA, that is 28S, 5.8S and 5S, and it contains 49 different proteins. So together these two makes ribonucleoprotein, and the small subunit contains only one ribosomal RNA, that is 18S plus 33 proteins, and together they make ribonucleoprotein. This is in case of eukaryotes, eukaryotic cells. So eukaryotic cell contains 2 ribosomal subunit, one large 60S, a small 40S. When these 60S combines with 40S give you a ribosomes of 80S. Large subunit contains 3 ribosomal RNA, 28S, 5.8S, 5S and 49 proteins. A smaller subunit contains only one ribosomal RNA, that is 18S plus 33 proteins. Similarly, if you will see the prokaryotic ribosomes, they contains 2 components, 50S and 30S. This 50-30 does not make 80, it makes 70S. And this 50S again contains 2 specific ribosomal RNA, 23S and 5S and 34 different proteins. And 30S contains only one protein, 18S, 21 proteins. So if you will see, compare it, see here, the difference between the prokaryotic and eukaryotic ribosomes is, eukaryotic ribosome is 80S, prokaryotic ribosome is 70S. The 2 subunit of 80S is 60 plus 40. In case of prokaryotes, it is 50 plus 30. And number of proteins and the number of ribosomal RNA also, it is differences. So this is overall picture. This is large subunit, this is a small subunit. Either it is prokaryotic cells or eukaryotic cells. Both have one large subunit, one a small subunit. And so here also you can see this is a small subunit and this is large subunit. Overall these two subunits joins together and they shares 3 specific locations, a specific place. That place we are going to call it sites. This is I don't know, it is A site. So 3 specific sites are there: A site, P site and E site. What is specific in this one is P and A. Remember, आपको कैसे याद करना, हाउ यू आर गोइंग टू रिमेंबर इट, आप कैसे याद करेंगे? A means it is called as amino acyl site, P site means peptidyl site and E means exit site. So first amino acid comes here. Whenever a amino acid will come, they will come here, and this place A site is present on both. थोड़ा पोर्शन इस पे भी होता है, थोड़ा पोर्शन इस पे भी होता है. Means the A site is going to be shared by large subunit as well as a small subunit. Similarly, P site is also shared by large and a small subunit. और यहाँ पे पहले सबसे पहले अमीनो एसिड आता है and peptide bond actually forms by this at this location. So this is called as peptidyl site या peptide binding site. And this is E site, that is maximum या solely belongs to the large subunit only. So ये small large subunit पे भी होता है और यहाँ से tRNA निकलता है, exit होता है. So that's why it is E site. This is here it is written there. So first site is A site, that is called as amino acyl site. So the admission of codon and messenger RNA charged, charge हम किसको बोलते हैं? मैंने बोला था कि tRNA विल बी लोडेड विथ एमिनो एसिड। हर एक एमिनो एसिड लोड होता है tRNA पे, अकेले आएगा नहीं। सो दिस इज कॉल्ड एज लोडेड tRNA या चार्ज tRNA। when a tRNA associated combined bind with amino acid, it is called as loaded tRNA, or you can call it charged tRNA. So first this charged tRNA comes to this A site, and that is present on large plus a small, both subunit. Second one is P site, that is peptidyl site. So peptide bond going to form here, and that is also on large plus a small. And third one is E site, which is mainly present on the large subunit only, and from here exit will be there, tRNA is going to exit. याद रहेगा इतना? Is it clear?
Yes, sir.
बाकी नहीं बोल रहे, बाकी others are there or not?
Yes, sir.
This is clear for everyone?
Yes, sir.
फाइन। जहाँ आपको कन्फ्यूजन लगे या समझ में ना आया, मुझे स्टॉप करेंगे तो मैं रुक जाऊँगा, बिकॉज मैं जब पढ़ा रहा हूँ ना तो मैं फुल स्क्रीन बोर्ड में होता हूँ, तो मुझे दिखता नहीं है कि अगर आपको चैट भी करेंगे तो, मैसेज भी करेंगे तो। सो प्लीज़ स्पीक, उसमें आप बोलिएगा तो ज़्यादा अच्छा रहेगा, ठीक है? So this is the basic difference between the ribosomes of prokaryotes and eukaryotes. आप इजीली डिफ्रेंशिएट कर सकते हैं। व्हाट इज द बेसिक डिफरेंस? 80s राइबोसोम्स मेनली प्रेजेंट इन यूकैरियोट्स, 70s राइबोसोम्स मेनली प्रेजेंट इन प्रोकैरियोट्स, माइटोकॉन्ड्रिया और क्लोरोप्लास्ट ऑफ यूकैरियोट्स। 80s जो बने होते हैं दो चीज़ों का, 60 एंड 40s का, 70s बने होते हैं दो का, 50 प्लस 30s का। 80s composed of 40% RNA and 60% protein. 60s is composed of 3 ribosomal RNA, 49 protein. 40s बने होते हैं एक ribosomal RNA and 33 proteins. इसी तरह से 70s will have 60% RNA and 40% protein. 50s will have 2 ribosomal RNA and 34 proteins, जो ऑलरेडी मैंने दिखा दिया आपको. 30s contains 16s ribosomal RNA and 21 different proteins. So this is some of the information about the ribosomes. So this is A site. So सबसे पहले amino acid यहाँ आएगा. This is P site which is present on the large and small subunit. Peptide bond यहाँ पे बनेगा. And this is E site, exit site. From here this जो आप बोतल जैसे यूनिट देख रहे हैं, that is the tRNA. So this tRNA जो without amino acid होगा, ये यहाँ से exit होगा. So this is called as E exit site, E site. And these are different codes. This code is going to be recognized by this tRNA anticodon, जो हमने कल भी देखा था. हर एक tRNA पे एक anticodon होता है. This anticodon is complementary to this genetic codes. So accordingly this tRNA will come here at the A site and bind it. तो आपका राइबोसोम जो है, ये जो व्हीकल देख रहे हैं राइबोसोम्स, मैसेंजर आरएनए नहीं मूव करता है, ये याद रखिएगा. व्हेन वी आर गोइंग टू डू द प्रोटीन सिंथेसिस, दैट टाइम मैसेंजर आरएनए मूव नहीं करेगा. मूव कौन करेगा? इट इज राइबोसोम विच इज गोइंग टू मूव. ठीक है. सेकेंड इंपॉर्टेंट कंपोनेंट जो, या उसमें लास्ट आप बोल दीजिए, जो हम कंपोनेंट जो हमारी जरूरत थी, दैट वाज टीआरएनए, ट्रांसफर आरएनए. इन ट्रांसफर आरएनए का काम क्या होता है? इट इज ट्रांसफर आरएनए इज गोइंग टू सर्व एज द कैरिंग द एमिनो एसिड टू द ए साइट ऑफ राइबोसोम्स. सो दिस टीआरएनए इज गोइंग टू बी लोडेड विथ amino acid. So each tRNA is charged with a particular amino acid at the third 3' end. अब RNA है तो obviously one end will be 5' end, other end will be 3' end. So at the 3' end it is going to join with a amino acid. रफली जो भी tRNA हमारे पास होते हैं वो होते हैं 73 to 93 amino न्यूक्लियोटाइड के, मतलब 100 न्यूक्लियोटाइड से नीचे होते हैं और 70 न्यूक्लियोटाइड से लंबे होते हैं. सो रफली ऑन एन एवरेज इट इज 90 to 93 न्यूक्लियोटाइड्स के होते हैं. दिस टीआरएनए, इफ यू सी, दिस इज द स्ट्रक्चर ऑफ टीआरएनए. So they are having, this is secondary structure. Secondary structure means planar structure, 2-dimensional structure. This is 3-dimensional structure. In 3-dimensional structure, it slightly looks like L-shape structure. When we are talking about 2D structure, it looks like clover leaf shape structure. अब जब आप tRNA देखते हैं तो इसमें मैं आपको, these are the specific area of the tRNA one by one. So first, this is 5' terminal, 5' phosphate. You can see here 5' phosphate. Second thing is this portion, because this portion is going to receive the amino acid, so it is called as acceptor stem, acceptor loop. Then we are going to have a CCA tail. On 3' side we are having for every tRNA, वो सिर्फ एक नहीं, सभी tRNA में CCA amino acid will be there. So on this adenine portion we are going to have amino acid will be joined here. So on this terminus we are going to have amino acid. So this is called as CCA tail or amino acid acceptor portion. Then we are having 3 important loops, that is D arm, which you can see here. Then we are going to see the T arm, T size C arm or T psi arm, and then this is anticodon. This is most important. 2 portions are important in this tRNA, that is one is CCA loop and second one is anticodon. ये तो आपका हो गया स्पेसिफिक थिंग्स. अब these tRNA are composed of nucleotides. आपने जो स्टार देख रहे हैं यहाँ G स्टार, this is one of the modified nucleotide. This is not the normal guanine, it is a modified guanine. Similarly, you can say psi, this is again a modified nucleotide. T, normally T means thymine. RNA में thymine normally नहीं होता है, but tRNA have certain unusual characteristic where tRNA contains some modified amino nucleotides, some kind of thymine also, and सेल्फ कॉम्प्लीमेंट्री रीजन्स. यहाँ देखिए दिस इज सेल्फ कॉम्प्लीमेंट्री. सो एक ही आरएनए का कुछ पोर्शंस ज्वाइन कर रहा है एक दूसरे से. सो दिस इज सेल्फ कॉम्प्लीमेंट्री रीजन्स. ठीक है. इन केस ऑफ ह्यूमन या इन केस ऑफ यूकैरियोट्स you can call it, roughly we are having around maximum 50 tRNA we can have in case tRNA. In case of human or eukaryote we can have up to 50 tRNA, and when we are talking about prokaryotic systems we are having 33 to 40 tRNA. And how many genetic for every means, if you are having 50 tRNA means how many anticodon loops I am having? 50 only. But how many genetic codes we are having? 61. So we require minimum 60 tRNA. हमारे पास 60 तो है नहीं, तो इसी को कंपेंसेट करने के लिए we are having vowel hypothesis या vowel means pairing. Clear?
Yes sir.
एक और important चीज, when we are talking about prokaryotic system, the messenger RNA are polycistronic. आप लिखना चाहें एक in case of prokaryotic system the mRNA are polycistronic, and in case of eukaryotic system it is monocystonic. Polycistronic means a single mRNA have many coding regions and which codes for many polypeptides. Many proteins can be synthesized from a single messenger RNA, and monocystonic means one mRNA will give you only one polypeptide, only one type of polypeptide, single polypeptides. In case of prokaryotic system, translation commences before the transcription of the gene is going to be complete. Means ट्रांसक्रिप्शन कंप्लीट नहीं हुआ बट mRNA जो आधा बना हुआ है वो ट्रांसलेशन करना शुरू कर देता है. सो इन प्रोकैरियोट्स ट्रांसक्रिप्शन एंड ट्रांसलेशन अकर्स टुगेदर. before transcription is going to completed, mRNA participate in translation process also. Whereas in case of eukaryotic system, because we have two different regions, one is nucleus जहाँ ट्रांसक्रिप्शन होना है, साइटोप्लाज्म में ट्रांसलेशन होना है, तो दोनों साथ साथ होता नहीं है, अलग अलग होता है. ठीक है. अब जो 5 रीजन्स हमने पढ़े हैं टीआरएनए में, 5 प्राइम टर्मिनल फॉस्फेट ग्रुप, इसकी बहुत ज़्यादा सिग्निफिकेंस नहीं है. एक्सेप्टर आर्म which receives the amino acid, that's why we are going to call it acceptor arm. CCA tail, that जो 3 प्राइम एंड पे CCA एंड होता है, which receives the TR-MR means amino acid molecules. So this is actual portion which receives amino acid. D arm, anticodon arm, and T arm, T size C arm or T arm. So these are the different modified nucleotides we can see in the tRNA. आप नॉर्मल एडेनिन, यूरासिल, साइटोसिन, गुआनिन के अलावा यू कैन सी ऑल दी टाइप ऑफ मॉडिफाइड न्यूक्लियोटाइड्स. 5 मीन्स स्यूडो यूरिडीन. आई अगर आप कहीं देखेंगे तो आई मीन्स इट इज आइनोसिन. आई यू कैन सी इन सम प्लेसेस इन केस ऑफ एंटीकोडोन लूप. याद कीजिए कल हम बात कर रहे थे वोवल हाइपोथेसिस में. If you are having inosine, it can pair with G, it can pair with A, it can pair with different other things. Then T, ribothamidine. D, 5, 6 means all are modified things. This is M1I, 1-methyl, M1G, 1-methyl guanosine, M2G, 2-methyl guanosine. So different type of modified nucleotides you can see in tRNA only, not any other RNAs. And this is our wobbling. So when you had in the anticodon tRNA you are having cytosine, it can pair only with G. If you are having anticodon A at the 3rd place, I am talking about, only it binds with U. If you are having U, it can pair with A also, it can pair with G also. If you are having G, it can pair with C also, it can pair with U also. And in case of inosine, it can pair with A, U and C. So this already we have seen during the vowel hypothesis or vowel pairing. यहाँ तक क्लियर है. सो दिस वाज द रिक्वायरमेंट्स. अभी तक हम जो देख रहे थे दैट इज द प्रिपरेशन ऑफ प्रोटीन सिंथेसिस. हमें क्या क्या चाहिए होता है, उसकी सिग्निफिकेंस क्या होती है. क्लियर.
Yes, sir.
किसी का कोई क्वेश्चन है? इफ यू हैव एनी क्वेश्चन यू कैन आस्क. If anyone is having any kind of questions, you can ask me. Okay. 2 चीज़ और याद कर लीजिए. इन विच डायरेक्शन डीएनए सिंथेसिस अकर्स. डीएनए सिंथेसिस किस डायरेक्शन में होता था?
5 prime to 3 prime, sir.
5 प्राइम टू 3 प्राइम, राइट. एंड आरएनए सिंथेसिस
So same 5 prime to 3 prime.
सेम. आरएनए और डीएनए ऑल्सो एंड आरएनए ऑल्सो. प्रोटीन सिंथेसिस भी आरएनए पे जो जाएगा वो 5 प्राइम टू 3 प्राइम ही जाएगा. मीन्स we are going to start reading the messenger RNA from 5 prime side of the RNA and moving towards 3 prime side. ठीक है. प्रोटीन का जो चेन बढ़ेगा वो बढ़ेगा आपका N टर्मिनस इंड से C टर्मिनस इंड की तरफ. सो एमिनो टर्मिनस टुवार्ड्स C टर्मिनस. ये सब याद कीजिएगा. दीज आर द डायरेक्शंस. DNA synthesis 5 prime to 3 prime means every time a new nucleotide will come, it will bind at 3 prime end. In case of RNA also, every time new nucleotide comes and bind at 3 prime end only. प्रोटीन सिंथेसिस के समय में मैसेंजर आरएनए की रीडिंग जो होगी वो भी 5 प्राइम टू 3 प्राइम डायरेक्शन में ही होगी. मीन्स जो सबसे पहला एमिनो एसिड होगा वो 5 प्राइम एंड पे होगा, पहला एमिनो एसिड. और सबसे लास्ट एमिनो एसिड जो जेनेटिक कोड्स जो रीड किया जाएगा दैट विल बी टुवार्ड्स द 3 प्राइम एंड. जो चेन प्रोटीन का चेन हम बनाएंगे दैट विल बी फर्स्ट न्यूक्लियोटाइड विल बी टुवार्ड्स N एंड every time a new amino acid will come and it will be joined at the C terminus. So these are some of the directions. अब जैसे कि आपने देखा था कि प्रोटीन सिंथेसिस आर गोइंग टू डिवाइडेड इनटू 5 डिफरेंट स्टेजेज. फर्स्ट portion is activation of amino acid, second is initiation, third elongation, fourth is termination and then it will be released, and last one is post translational processing, modification or protein folding you can call it. So हम शुरू कर रहे हैं पहला, that is activation of amino acid. अब हर एक स्टेज के लिए हमें कुछ कुछ अलग अलग चीज़ों की ज़रूरत पड़ेगी. ये दिस टेबल इज फ्रॉम द लेहनिंजर. Leninger's biochemistry. So during activation of amino acid we require 20 amino acids. तो मैंने बोला कि हर एक अमीनो एसिड्स के लिए हमें क्या चाहिए, एक स्पेसिफिक एंजाइम. तो 20 अमीनो एसिड्स के लिए हमें कितने टीआरएनए सिंथेटेज चाहिए, 20 डिफरेंट अमीनो एसाइल टीआरएनए सिंथेटेज. अब ग्लाइसिन के लिए हमको चाहिए ग्लाइसाइल टीआरएनए सिंथेटेज, एलानिन के लिए चाहिए हमको एलालाइल टीआरएनए सिंथेटेज. सो this amino acyl can be replaced with the specific amino acids name with the ending with -yl, glycyl, allyl, phenylalyl, इस तरह से. 33 to 40 tRNA we are having in case of prokaryotes. In eukaryotes up to 50. So we require those tRNA also. हमें energy source चाहिए in the form of ATP. And whenever we are going to consume energy we require magnesium ion. So this is our requirement for the activation of amino acid. अब एक चीज़ और समझ लीजिए, in biochemistry we are going to use two terms. One is we are going to call it synthase and second term we are going to use synthetase. Can anyone will tell me the difference between synthase and synthetase, or both are same? Is it same thing or is it different?
So antagonistic.
No. Anyone else? और कोई? Both are going to catalyze the synthetic reaction. Synthetic means जब हम कुछ बनाने लगते हैं तो हम synthesis करते हैं, synthesize करते हैं. तो in the either synthase or synthetase both are same thing, same enzyme which is going to produce certain things. वो सिंथेज रिएक्शंस जहाँ आप कुछ सिंथेसाइज करते हैं और हमें एनर्जी की ज़रूरत पड़ती है. सो दोज सिंथेज रिएक्शंस विच इज गोइंग टू प्रोड्यूस सर्टेन थिंग्स एंड रिक्वायर्स एनर्जी, तो उसको हम बोलते हैं सिंथेटेज. सो इट मीन्स every time wherever we are going to see the synthetase, it means this is a energy consuming synthesis process. It requires energy. ये भी एक तरह का सिंथेज ही है, बट सिंथेज से आपको ये पता नहीं चलेगा कि ये इसमें एनर्जी की ज़रूरत है या नहीं है. वो हो सकता है कुछ एनर्जी चाहिए, कुछ में नहीं चाहिए, बट जब भी हम बोलेंगे सिंथेटेज, इट ऑलवेज मीन्स दैट एनर्जी इज गोइंग टू बी कंज्यूम्ड देयर. ठीक है यहाँ तक?
Yes, sir.
फाइन. सो दिस इज आवर रिक्वायरमेंट फॉर द एक्टिवेशन प्रोसेस. इसके बाद जब हम एमिनो एसिड को एक्टिवेट करते हैं, इसका काम क्या है कि वी आर हैविंग ए टीआरएनए 30 और 40 और 50 वट एवर टीआरएनए वी आर हैविंग. amino acid will be loaded on this one. So the activation of amino acid means we are going to load amino acid on the CCA end of the tRNA. CCA end कहाँ होगा? 5 प्राइम एंड पे या 3 प्राइम एंड पे?
3 prime end.
3 प्राइम एंड पे. And in this CCA, which side amino acid is going to join? Towards A side or towards C side?
A side.
A side. And this CCA end is almost opposite to the anticodon loop. anticodon loop will be at this side, this end, and CCA end will be on opposite side. So after in the activation we have done for all the 20 amino acid is going to be loaded on their respective tRNA depending on the genetic codes. So we require different tRNA in different cases. When we are talking about initiation process, हमें सिर्फ एक अमीनो एसिड की जरूरत पड़ेगी एंड दैट इज द मिथियोनिन, बिकॉज नॉर्मली मिथियोनिन इज द फर्स्ट अमीनो एसिड विच इज गोइंग टू start the protein synthesis, because AUG is going to the initiation codon. ठीक है. So आपके मैसेंजर RNA पे AUG will be the first genetic codes which is going to be read by a ribosomal RNA or you can call it tRNA. So AUG means it is methionine. तो यहाँ हमें सबसे पहला अमीनो एसिड जो होगा वो मिथियोनिन बाइंड होगा. अब मिथियोनिन दो तरह के हो सकते हैं, या तो ये पहला मिथियोनिन होगा जो यहाँ पे होगा, या हो सकता है कुछ मिथियोनिन बीच में भी हो कहीं भी. इसके अलावा दिस इज द फर्स्ट अमीनो एसिड एंड दिस इज द मीडियरी और सेकेंड और थर्ड और फोर्थ और कहीं भी हो इसके अलावा, पहले के अलावा. तो दो तरीके का मिथियोनिन हो गया, फर्स्ट इनिशिएशन कोडॉन, इनिशिएशन मिथियोनिन और इंटरनल मिथियोनिन. दोनों को डिफ्रेंशिएट करने के लिए सेल्स के पास एक स्पेसिफिक प्रोसेस होता है इन विच दोज tRNA विच इज लोडेड विथ इनिशिएशन मिथियोनिन, जिसको सबसे पहले आना है, दैट मिथियोनिन इज गोइंग टू बी फॉर्माइलेटेड. तो इसको हम बोलते हैं FMET, फॉर्माइलेटेड. फॉर्माइल ग्रुप मीन्स COCH3, ये ग्रुप ऐड हो जाता है मिथियोनिन पे. So this is called as formyl group and the amino acid is called as formilated methionine. तो अब मिथियोनिन के लिए सेल के पास दो तरह की tRNA है, एक tRNA जिस पे फॉर्माइलेटेड मिथियोनिन है और एक tRNA जिस पे नॉर्मल मिथियोनिन है, ओनली मिथियोनिन नॉट फॉर्माइलेटेड. तो ये वाला tRNA जो है, जो सबसे पहला मैसेंजर RNA पे जो AUG होगा, अगर यहाँ AUG है तो यहाँ ये tRNA आएगा, और इसके बाद और कहीं AUG है तो ये वाला tRNA आएगा. सो ड्यूरिंग इनिशिएशन वी आर गोइंग टू यूज N-formyl methionine लोडेड ऑन tRNA. सो वी रिक्वायर दिस ड्यूरिंग द इनिशिएशन प्रोसेस, और इसकी जरूरत जो पड़ेगी वो पड़ेगी ड्यूरिंग इलोंगेशन. इसके अलावा वी रिक्वायर बोथ द राइबोसोमल सब यूनिट 30s एंड 50s इन केस ऑफ प्रोकैरियोट्स. इन केस ऑफ यूकैरियोट्स हमें 60 और 40, हम लेटर ऑन देखेंगे यूकैरियोट्स का, बिकॉज वो दैट इज लिटिल बिट कॉम्प्लिकेटेड, तो हम वो बाद में देखेंगे. पहले प्रोकैरियोट्स कर लेते हैं. सो 30 एंड 50s राइबोसोमल सब यूनिट, 3 इनिशिएशन फैक्टर्स, दैट इज इनिशिएशन फैक्टर 1, 2 एंड 3, एंड एनर्जी सोर्स GTP, एंड जहाँ एनर्जी जरूरत पड़ेगी वहाँ मैग्नीशियम. So this is our requirement for initiation step. Then during elongations we require, because initiation हम सिर्फ एक ही tRNA से करेंगे, उसके बाद जितने भी tRNA होगा that will comes during the elongation process. And elongation process के केस में हमें कुछ elongation factor की जरूरत पड़ेगी, that is called as elongation factors. So 3 elongation factor, EFTU, EFTS, EFG. इन आर केस, हमारे केस में 1, 2, 3 बोलते हैं इलोंगेशन यूकैरियोटिक इलोंगेशन फैक्टर 1, 2, 3. अगर हम ये नहीं भी यूज करेंगे तो EF1, EF2, EF3. ये यहाँ भी हमें हर एक tRNA जो आएगा वो GTP के साथ आएगा, तो GTP एंड बिकॉज एनर्जी चाहिए तो मैग्नीशियम. सो दिस इज ऑल्सो. एंड लास्ट प्रोसेस इज we require releasing factor or termination factor. So it is RF1, RF2, RF3 in case of prokaryotes. तो ये हमें प्रोटीन सिंथेसिस के लिए चाहिए. जब प्रोटीन बना लेंगे देन वी आर गोइंग टू मॉडिफाई आर प्रोटीन्स. दो तरीके का, एक प्रोटीन में हम कुछ उसको हम स्ट्रक्चरल मॉडिफाई करेंगे, जैसे हमने प्रोटीन बनाया सीधा, इसको हमने तोड़ जोड़ के टेढ़ा टोढ़ा करके एक 3 डायमेंशनल स्ट्रक्चर दे दिया. दिस इज कॉल्ड एज स्ट्रक्चरल मॉडिफिकेशन, कॉन्फॉर्मेशनल मॉडिफिकेशन और स्ट्रक्चरल मॉडिफिकेशन, जहां आपने स्ट्रक्चर को चेंज कर दिया. एंड दूसरा वी आर गोइंग टू डू द केमिकल मॉडिफिकेशंस, जहां हम कुछ स्पेसिफिक ग्रुप ऐड करेंगे, जहां फॉस्फेट, मिथाइल, कार्बोक्सिल, कार्बोक्सिलेट, प्रोस्थेटिक ग्रुप्स, ग्लूकोज मॉलिक्यूल्स, यूटीपी मॉलिक्यूल्स. सो केमिकल चेंजेज, so structural modification and chemical modifications. Is this clear? Activation, initiation, elongation, termination and modification.
Yes, sir.
Okay. So the first step of activation of amino acid. This for the synthesis of polypeptide with a specific sequence, हम specific sequence का polypeptide बनाना है. वो हमें इंफॉर्मेशन कहाँ से आएगा? मैसेंजर आरएनए से. उसके लिए हमें मिथियोन, सबसे पहला अमीनो एसिड हमें मिथियोनिन चाहिए, या दूसरे अमीनो एसिड हमें इलोंगेशन स्टेप में चाहिए, जिसको हमें लोड करना है टीआरएनए पे. तो फॉर द सिंथेसिस ऑफ पॉलीपेप्टाइड विद डिफाइंड सीक्वेंस, टू फंडामेंटल केमिकल रिक्वायरमेंट मस्ट बी मेट. द कार्बोक्सिल ग्रुप ऑफ ईच अमीनो एसिड मस्ट बी एक्टिवेटेड. अब आपने देखा था अमीनो एसिड का बेसिक स्ट्रक्चर. मैंने बोला कि हम प्रोटीन सिंथेसिस में कौन सा अमीनो एसिड यूज करते हैं अल्फा बीटा गामा में से?
अल्फा सर.
सो अल्फा में क्या होगा? दिस इज COOH ग्रुप, दिस इज NH2 ग्रुप. अल्फा कार्बन पे अमीनो ग्रुप होगा, एक हाइड्रोजन होगा, एक R ग्रुप होगा. दिस इज वन बेसिक अमीनो एसिड. तो दो चीज़ें हमें करने की जरूरत पड़ती है. जो हम एक्टिवेशन की बात करते हैं तो हमें दो चीज़ें पहले करनी चाहिए. पहला, ये COOH ग्रुप जो है, दिस मस्ट बी एक्टिवेटेड. सो द कार्बोक्सिल ग्रुप ऑफ ईच अमीनो एसिड मस्ट बी एक्टिवेटेड. इसको हम ऐसा तैयार कर लें सो दैट इट विल इजीली फॉर्म्स द पेप्टाइड बॉन्ड. एंड सेकेंड इज, ए लिंक मस्ट बी इस्टेब्लिश्ड बिटवीन ईच अमीनो एसिड एंड द इंफॉर्मेशन ऑफ द मैसेंजर आरएनए. अब ये अमीनो एसिड जाएगा मैसेंजर आरएनए पे, वो कैसे पता चलेगा? विद द हेल्प ऑफ टीआरएनए. तो इसके लिए हमें इस अमीनो एसिड को लोड करना पड़ेगा टीआरएनए पे. सो पहला मॉडिफिकेशन ऑफ COOH ग्रुप, and second, this amino acid should be loaded on tRNA. So both these requirements जो हम ये भी पढ़ रहे हैं, met by the attaching the amino acid to a tRNA in the first stage of protein synthesis. Attaching the right amino acid to the right tRNA is very very critical. कि सही, हमारे पास 20 अमीनो एसिड हैं, 61 तरह के जेनेटिक कोड्स हैं, एंड रफली 40 to 50 टीआरएनए है. अब इस 20 अमीनो एसिड को 40 टीआरएनए पे लोड करना अकॉर्डिंग टू दिस 61 जेनेटिक कोड्स. तो यहाँ पे बहुत क्रिटिकल पॉइंट्स होता है कि आपको सही टीआरएनए पे सही अमीनो एसिड लोड हो अकॉर्डिंग टू द सही जेनेटिक कोड्स. ठीक है. और ये रिएक्शन जो एक्टिवेशन हो रहा है, ये साइटोप्लाज्म, फ्री साइटोप्लाज्म हो रहा था, ना कि राइबोसोम्स पे. तो दिस इज, यू कैन कॉल इट वन ऑफ द वेरी इंपॉर्टेंट स्टेप. जहाँ सही अमीनो एसिड ऐड होगा प्रोटीन में, उसके लिए ये स्टेप सही होना जरूरी है. इन दिस केस, फॉर द ईच 20 अमीनो एसिड्स कोवैलेंटली एसोसिएटेड ऑन द टीआरएनए एट द एक्सपेंस ऑफ ATP. आपने देखा था ATP की जरूरत पड़ी थी मैग्नीशियम आयन के प्रेजेंस में, एंड हमें एक एंजाइम चाहिए था, दैट इज एमिनो एसाइल टीआरएनए सिंथेटेज. Again, amino cell tRNA synthesis in specific to amino acid. अब हमारे पास 50-40 tRNA है, तो हमारे पास 40 एंजाइम नहीं है, हमारे पास 20 अमीनो एसिड के लिए 20 एंजाइम है. तो एक अमीनो एसिड अगर दो तरह के टीआरएनए पे लोड हो रहा है, तो एक ही एंजाइम काम करेगा. तो दिस एंजाइम इज स्पेसिफिक टू अमीनो एसिड, नॉट स्पेसिफिक टू टीआरएनए, नॉट स्पेसिफिक टू जेनेटिक कोड्स. ये रिमेंबर की, ये याद कर लीजिए, अंडरलाइन कर लीजिए. एंड अमीनो एसाइल टीआरएनए सिंथेटेज, इट इज ए कॉमन नेम फॉर एवरी अमीनो एसिड. वी कैन हैव ए स्पेसिफिक नेम. ठीक है, जैसे ग्लाइसाइल, एलालाइल, इस तरह से नाम हम कर सकते हैं. During the first stage of protein synthesis, amino acid tRNA synthetase strify the 20 amino acid at their corresponding tRNA. आप पहले रिएक्शन देख लीजिए, फिर हम बात कर लेते हैं. This is the reaction. This reaction occurs in two steps. The first step is amino acid is going to react with ATP. ATP का AMP पोर्शन, मोस्ट ऑफ द केसेस हम AMP पोर्शन यूज करते हैं, तो यहाँ का AMP is going to bind with this amino acid in the first step. So we are going to have amino acyl AMP, and यहाँ से दो फॉस्फेट हमने निकाल दिया, बीटा एंड गामा फॉस्फेट. So this is my first reaction product. In the second step, this component is going to combine with tRNA. So AMP will release, AMP release हो जाएगा, और this amino acid will be loaded on tRNA. So ultimately you have got amino acylated tRNA. This is the chemical reaction. यही करवाना है हमें during activation process. This is the chemical reaction. This is your amino acid where we are having COOH group. This is central carbon alpha carbon. This is hydrogen. आप इसको इस तरह से लिख सकते हैं, this hydrogen को. This is amino group. This is R group. In the first step, this is the enzyme. ATP is going to involve. यहाँ से बीटा एंड गामा फॉस्फेट को हमने हटा दिया. AMP we will use, and AMP will associated with this enzyme adenosyl pyrophosphate, and this is the from amino acid. In the second stage हमें tRNA चाहिए. So this tRNA joins with this portion. So ultimately you are going to have tRNA loaded with amino acid. So दो स्टेज में, फर्स्ट स्टेज ATP consumes and it will give AMP to the amino acid. We will get amino acyl ATP, and second stage, sorry, amino acyl amino acid, क्या बोलते हैं, amino acyl AMP, and in the second stage it will combine with tRNA. AMP portion will be released and tRNA will loaded with amino acid. So this reaction occurs for all the 20 amino acids. ये हर एक अमीनो एसिड्स के लिए होना है. दिस इज द सेम प्रोसेस. अमीनो एसिड ATP, दो फॉस्फेट निकल गया, AMP पोर्शन इज जॉइंट्स विद द COOH ग्रुप ऑफ अमीनो एसिड. सो दिस इज एडिनाइलेटेड अमीनो एसिड. इन द सेकेंड स्टेज दिस इज द tRNA. दिस पोर्शन इज एंटीकोडोन लूप. दिस इज CCA. अब ये यहाँ OH क्यों दिखा रहा है? बिकॉज दिस इज द 3' एंड, एंड 3' एंड पे OH होते ही हैं. So this is the 3' end OH of adenine. So this amino acid from this place, it is going to be transferred to the OH group of adenine at the 3' CCA, and amino acyl AMP will be released. So this is loading of amino acid on tRNA, and for every amino acid this process will be repeated. Clear? सेम रिएक्शन आप देख सकते हैं अलग अलग जगह पे, अलग अलग तरीके से. दिस इज अमीनो एसिड, दिस इज कार्बोक्सी टर्मिनल, दिस इज ATP. सो दिस पोर्शन इज गोइंग टू बी कंज्यूम्ड हियर, एंड दिस पोर्शन विल बी फ्री in the form of pyrophosphate. So this is going to combine with this one. So this is your amino acyl AMP. In this case, in the next reaction tRNA will be utilized. This portion will be transferred to the tRNA. This is here, this is adenine, CCA का adenine, and this is 3' OH. On this portion, this group amino acid has been acylated. So this is amino acylated tRNA और loaded tRNA और charged tRNA, जो भी आप बोलना चाहें बोल सकते हैं. It is same thing. ठीक है, यहाँ तक क्लियर?
Yes sir.
Anyone is having any confusion? अब मैं थोड़ा कॉम्प्लिकेट करने वाला हूँ, इसलिए अभी तक कोई प्रॉब्लम है तो बताइए. इफ यू हैव एनी कंफ्यूजन यू कैन आस्क मी. नहीं, ठीक है. अब जो आप पिक्चर यहाँ देखने वाले हैं, दिस इज द फर्स्ट रिएक्शन ऑफ द एक्टिवेशन प्रोसेस. In this activation process, we are going to have, this is the amino acyl दिस एमिनो एसिड इज लोडेड ऑन AMP पोर्शन. जो हमारा एंजाइम होता है एमिनो एसाइल tRNA सिंथेटेज, दैट इज ऑफ टू टाइप्स. वी आर हैविंग टू क्लासेस ऑफ एमिनो एसाइल tRNA सिंथेटेज. दोनों में बेसिक डिफरेंस क्या होता है? हाउ दे आर गोइंग टू इन्वॉल्व इन द रिएक्शन, हाउ दे आर गोइंग टू पार्टिसिपेट इन द रिएक्शन. सो वी आर हैविंग क्लास 1 एंड क्लास 2. थोड़ा कंसंट्रेट होकर देख लीजिए, ध्यान दे दीजिए. आई विल स्टॉप हियर ओनली. इसके बाद so in case of class 1 reaction, when we are talking about class 1 enzyme, so this is your amino acid loaded on joints with AMP after the first reaction. So this is amino cell tRNA. अब आप जानते हैं कि CCA की मैं बात कर रहा हूँ. in this is CCA, this is the last nucleotide of tRNA. सो दिस एडेनिन का 3 प्राइम पे भी OH है एंड सेकेंड प्राइम पे भी OH है, बोथ बिकॉज दिस इज RNA, सो बोथ OH इज फ्री. ऑन दिस एडेनिन वी आर हैविंग 2 प्राइम OH ऑल्सो एंड वी आर हैविंग 3 प्राइम OH ऑल्सो बिकॉज इट इज RNA. अगर रिएक्शन में जो लोन पेयर, दोनों ऑक्सीजन के पास लोन पेयर है, इफ द लोन पेयर ऑफ सेकेंड ऑक्सीजन, सेकेंड प्राइम ऑक्सीजन का लोन पेयर पार्टिसिपेट करे रिएक्शन में, तो क्या होगा? सबसे पहले सेकेंड पोजीशन का लोन पेयर इलेक्ट्रॉन अटैक करेगा. सो एमिनो एसिड विल बी फर्स्ट ट्रांसफर टू द सेकेंड पोजीशन ऑफ द एडेनिन, जो आप यहां देख रहे हैं. एंड देन इन द सेकेंड नेक्स्ट रिएक्शन इट नीड टू बी ट्रांसफर टू द थर्ड प्लेस. सो उसका next reaction, this lone pair will attack and this amino acid will be ultimately transferred to the third place. So last में मुझे ये थर्ड पोजीशन पे ही चाहिए. This amino acid we require at the third position of oxygen, but in the second stage if it is going to be mediated by the second prime oxygen, तो उसको हम बोलते हैं tRNA synthetase enzyme type 1. So type 1 enzyme involve second stages in 2 steps. पहले में सेकेंड प्राइम ऑक्सीजन पार्टिसिपेट करेगा रिएक्शन में, देन 3 प्राइम का ऑक्सीजन पार्टिसिपेट करेगा रिएक्शन में. सो 2 स्टेजेज में सेकेंड रिएक्शन होगा इन केस ऑफ टाइप 1 एंजाइम. व्हेन वी आर गोइंग टू टॉक अबाउट टाइप 2 एंजाइम, इन दैट केस this portion directly third oxygen is going to involve in the reaction. So only one step and directly it will be this amino acid will be transferred to the third step, third OH. Clear? Is this okay?
Yes, sir.
Yes, sir.
So we are having two types of tRNA synthetase where it is going to be, if first stage is common in both the cases. During the first stage amino acid combines with ATP gives you amino acyl 8 AMP, जहाँ amino acid loaded होगा ये पोर्शन ऑन AMP. सेकेंड स्टेज में जब हम tRNA पे लोड करते हैं वहाँ डिफरेंस आता है कि अगर वो मीडिएटेड बाय सेकेंड प्राइम OH देन ट्रांसफर टू द 3 प्राइम OH, तो उसको हम बोलते हैं टाइप 1 एंजाइम, और टाइप 2 एंजाइम डायरेक्टली ट्रांसफर टू द 3 प्राइम OH ओनली. तो देर आर सर्टेन स्पेसिफिक एमिनो एसिड्स जिसके लिए टाइप 1 होते हैं, सर्टेन स्पेसिफिक एमिनो एसिड जिसके लिए टाइप 2 का प्रोसेस होता है. अगर आप लिखना चाहें तो लिख लीजिए. टाइप 1 की मैं बात कर रहा हूँ पहले. टाइप 1 आर्जिनिन, सिस्टीन, ग्लूटामीन, isoleucine, leucine, then we are having methionine, I think methionine, then tryptophan, then we are having tyrosine, then we are having valine. So 3 4 5 हाँ, for this 10 amino acids we are having type 1 enzyme which is mediated by second prime OH then to third prime OH. So 2 स्टेज में दूसरा रिएक्शन होगा. एंड टाइप 2 एंजाइम जो होगा दैट इज फॉर एलानिन, एस्पेराजीन, aspartic acid, glycine, histidine, lysine, phenylalanine, proline, serine, and threonine. This is histidine. So for these 10 amino acids we are having type 2 enzymes. ठीक है यहाँ तक? Any confusion? Anyone have not understood this reaction, this process or anything yet? Please tell me. अब तो खत्म होने वाला क्लास है, आप तो बोल दीजिए यस और नो.
क्लियर.
आप ही बोलते हो, बाकी तो कोई बोलते नहीं है. चलिए. सो ये दिस इज द लिस्ट. दिस इज फॉर फर्स्ट क्लास, दिस इज फॉर रिमेनिंग सेकेंड क्लास. आप स्नैपशॉट लेना है तो ले सकते हैं. ठीक है. Now this stage amino acyl tRNA synthetase is doing some kind of proofreading also. Proofreading means मैंने बोला था कि ये स्टेज बहुत क्रिटिकल स्टेप होता है. क्या क्रिटिकल स्टेप होता है? कि सही एमिनो एसिड सही tRNA पे सही जेनेटिक कोड्स के अकॉर्डिंगली लोड हो. सो दैट इज proofreading. So the amino acylation of tRNA accomplishes two ends. First, activation of amino acid for a peptide bond formation and attachment of amino acid to the adapter tRNA ends, that is CCA end, that ensures the appropriate placement of amino acid for a growing polypeptide. To identify the amino acid attached to tRNA is not checked on the ribosomes. अब आपने tRNA पे कौन सा amino acid लोड किया है ये राइबोसोम कभी चेक नहीं करता है. आपने सही एमिनो एसिड लोड किया है, मान लीजिए यहाँ AUG है, बट लोड आपने मिथ्योनिन के बदले में कुछ और भी कर दिया है, तो राइबोसोम्स इज नॉट गोइंग टू चेक इट. सो दैट शुड बी डन एट दिस लेवल ओनली. राइबोसोम्स नेवर गोइंग टू चेक दैट tRNA कम्स विद द करेक्ट एमिनो एसिड और रॉन्ग एमिनो एसिड. सो द अटैचमेंट ऑफ द करेक्ट एमिनो एसिड टू द tRNA इज इसेंशियल फॉर द fidelity of protein synthesis. Fidelity means सहीनेस होने के लिए. So the first filter, पहला जो इसका प्रूफ रीडिंग होता है, that is the initial binding of amino acid to the enzyme and its activation to amino acyl AMP, जो आपने यहाँ रिएक्शन में देखा था. So the first filter is this process, this reaction, कि amino acid react करेगा AMP के साथ, ATP के साथ and give you amino acyl AMP. And second filter is binding of any incorrect amino acyl AMP product to separate active site of an enzyme. A substrate that binds to the second active site is hydrolyzed. अगर आपने गलत तरीके का एमिनो एसाइल AMP बना लिया और एंजाइम है किसी दूसरे tRNA सिंथेटेज का और बाइंड कर गया आपके पास दूसरा एमिनो एसिड, तो उस केस में दिस शुड बी हाइड्रोलाइज्ड. इसको हम डिग्रेड कर देते हैं, इसको प्रोसेस नहीं करते हैं फॉर बाइंडिंग टू द tRNA. सो दो स्टेज में ये प्रूफ रीडिंग होता है. पहला सही एमिनो एसिड एक्टिवेट हो और दूसरा सही एमिनो एसिड लोड हो tRNA पे. अगर ये गलत होता है तो एमिनो एसाइल tRNA सिंथेटेज उसको हाइड्रोलाइज कर देता है. इट इज नॉट गोइंग टू प्रोसेस फर्दर. सो दिस इज कॉल्ड एज प्रूफ रीडिंग डन बाय एमिनो एसाइल tRNA सिंथेटेज. ठीक है.
Yes, sir.
So I am going to stop it here today. Tomorrow we will do the elongation, means initiation. Actual initiation क्या होगा? अभी तक हमने सिर्फ activation देखा, initiation, elongation and termination. That we will see tomorrow. Okay. आपके पास कुछ क्वेश्चन है तो पूछ सकते हैं. If you have any kind of questions, you can ask.